Hi, today we talk about diabetes, not in a pathophysiological point of view and not in a medical view because it's boring. Everybody talks about diabetes, and we will talk about it in a different way. Okay, with the focus always of the central dogma, and we will try to relate the central dogma to all the world around diabetes. You know, the biotechnological point of view, the research point of view, and we will see. Okay, so we will just briefly summarize what diabetes is in order to understand what is the world around diabetes. So diabetes type 2 is just a problem with the system of insulin. Insulin is an hormone secreted by these glands. It's called pancreas, this red bean. This is a slice of pancreas, and there are these cells in the pancreas that produce this hormone. This hormone is a very particular hormone because it's also a protein, and if insulin is a protein, these cells must have into their nucleus a DNA chunk that code for an RNA that code for insulin protein. So in the case of diabetes, what happens is that these cells sometimes just stop to make insulin, and this is a problem because insulin. The role of insulin is to flow in the blood and to make your cell of the body capable of grabbing the sugar from the blood. So if these cells stop to make insulin, your cells of your entire body will stop to take glucose and sugars from your blood. So the sugar on the blood will accumulate, and it's a problem because at a very high concentration glucose is dangerous, and it will be excreted into your urine. By the way, this is the reason it's called diabetes mellitus. Mellitus, it means in Latin, it means honey. Mellitus, it's like sweet like honey, because there was a time when the doctors, there wasn't a lot of the diagnosis tool like today. They drank the urine from the patients, and the urine of the people with the diabetes were very very sweet like honey, because if the sugar it's not taken by your cells, it goes away. So this is diabetes. Now let's look at what is the approach to treat diabetes. Okay, so initially they found that diabetes was a carence in insulin production, and they thought, the scientists, the doctors thought, if this pancreas of this person doesn't make insulin, let's take the insulin from another body, from another people. So the first approach to treat diabetes was, we take the insulin from the pancreas of this other man, and we will cut it. We will purify the insulin into these cells, because into these cells of healthy people will be some insulin, so we will purify it. We will put it into a syringe, and the patients could shot it into their bellies. So there is just one problem. The problem is that to extract the insulin from pancreas, you have to take the pancreas. It's like inside you. So you have to wait for these people to die. The healthy people must die before they could donate their insulin. And for a first approach is good. It worked, but it's really expensive. It's low because you have to wait for people to die, and it's not very efficient. So, but researchers and doctors, were smart, and they thought, insulin is a protein. So it's made in the pancreas. We are mammals. What? Why can't we take the insulin from another mammals, like a pig? Okay. So the next approach to treat diabetes in patients was to take the insulin from the pancreas of pigs. So this was slightly more efficient and also less expensive, because you can operate your pigs every day when you want. Pigs doesn't say anything. You can open them and take the insulin, take a piece of their pancreas and purify the insulin and fill a syringe, and so on and so forth. And this was a lot more efficient than the first procedure, but there is another problem. There is a different problem. The problem is that we said that insulin is a protein, and we said that the protein is coded by the RNA that is coded by the DNA. Now, the pig's DNA and they are nowadays different from the DNA of the human. So the insulin protein of the pigs were different from the insulin proteins of the human, and when you purify it and inject it in patients, sometimes the immunitary system goes crazy, because it's not a protein that the immunitary system recognizes as a human protein. So there was this problem, and also it was a little bit expensive, because you have to purify it, and it's not so easy. You have to operate your pigs, and it's not a definitive treatment. It's not a definitive process. I mean, you are taking pig's pancreas. It's not very perfect. So, but researchers and doctors were smart, and they thought they got back to the basics. Okay, they've gone back to the central dogma of molecular biology, and they thought insulin is a protein, and if insulin is a protein, there is an RNA that code for this protein, and the RNA is coded by a DNA. So we don't need an entire human organism or a mammals to produce insulin protein. We just need the informational content in the DNA. Okay, so how beautiful if we find the chunk of information for insulin protein, and we can isolate it so that we can have just the information for protein, and we can make it whatever we want with it. So this is what they've tried to do, and they isolated the chunk of DNA for insulin in the human genome. Okay, so this rectangle is the insulin chunk of DNA, okay, in the human DNA, in the human sequence of nucleotides. So this is the protein, this is insulin, but in order to express the potential of the information content of the DNA, you need all a gigantic apparatus. You need the nanomachine, remember, the RNA polymerase, and you need ribosome. You need what's called a vector of expression. You need an apparatus that can exploit the potential of the information into the DNA. And what apparatus do you think will fit our need? They have thought about bacteria. Okay, bacteria are perfect vector of expression because their growth is very fast. They are cheap, and you can take tons of bacteria into some fermenter and just let them go. They don't make noise. They are just perfect. So they cut the chunk of DNA that code for the insulin protein with some genetic scissors. We will talk about it maybe in other videos. They have cut the DNA, they have transposed the informational content for insulin into the bacteria, and now the bacteria can produce insulin. So you just put your bacteria into some gigantic fermenter, they grow, they eat, and they produce a lot of insulin. Now you just have to kill some bacteria, and it's really easy. Also in an ethic fashion, you know, they don't scream, they don't bleed. It's really easier to kill the bacteria, and also to extract some protein. So you produce your protein in a bacteria vector. Okay, so this is it for today. It's just another example of how you can use the central dogma of molecular biology. You know, this is possible just because we know that a chunk of DNA is all is needed to code for a protein. Okay, so whatever, for all the protein that you are searching for, if you find your chunk of DNA, you will successfully produce a lot of your protein. I mean, this is awesome. Okay, this saved a lot of lives, and today diabetes is no more a difficult disease to treat, just because of some researchers and some doctors have gone to the basics of the central dogma of molecular biology. So this is it for today. Let me know, what do you think about it? What do you think will be the next step on this process of treating the diabetes disease? And let me know if this was helpful, and see you later. Thank you for watching.